So, Jack and I are going to teach you about meiosis. Oh, he's not as oh, no, he's just showing up. Okay, I'm going to teach you about how to draw and model it. So make sure you have your modeling meiosis paper out with you. And you're going to draw along with me as I present it to you with these little pot bead things. They pop out and come back together, which is why they're called pot beads. So, the purpose of meiosis, you should have watched the Amoeba Sisters video. If you haven't already, please go do that. But the purpose of meiosis is to create genetic diversity when we're making egg and sperm cells. So when you make egg and sperm cells, you want each one to be unique, and that's what we're going to have in meiosis. We're going to have unique cells come out at the end of this. However, because we're making egg and sperm cells, keep in mind that we also, if there's a mutation that occurs in this process, then those mutations are passed down to offspring potentially, if that sperm and that egg were to become a baby. So this is where mutations can be passed down to children, only in meiosis when you're making egg and sperm cells. Okay, so we're going to start out with two different sets of chromosomes. So you can call this one chromosome 1, and then we have a copy from mom and a copy from dad. And chromosome 2, we have blue's going to be dad and red's going to be mom. And one thing to keep in mind as we're going through this, we're going to generate some genetic diversity. The goal of meiosis is to be different. You want each of your egg and sperm cells to be genetically different, which is why you and your siblings don't look the same, because you weren't made from the same egg and sperm cells unless you're identical twins, then you were. But if you weren't, then this is why. Okay, so it starts out very similar to mitosis. And in fact, some of these phrases are going to be the same, but it's going to be slightly different in a few ways. So the first one is interphase. And this is similar to mitosis interphase. In interphase, we have, they're just hanging out. They're going to signal to divide to make sperm and egg cells. So we need to do DNA replication, just like we do before mitosis. So we need to replicate our DNA. Wow, look at that. We went from these individual chromatids to now these replicated chromosomes, okay? So each of these got an identical strand, it just replicated DNA. So these two strands are the same. These two strands are the same. These two chromatids are the same. And these two are the same. They're identical because all we did was replicate DNA, just as we know it occurs. Okay, so now that's what you should have for interphase. So for interphase, you're just going to draw this, and you could put, if you want, this is chromosome type 1, and then these are type 2. We have 2 sets of chromosomes. All right, prophase and interphase are going to be very similar, okay? So prophase is going to, basically in interphase, we've replicated the DNA. We've gone from the 1 strand to 2 strands of each type of chromosome, okay? So prophase is going to look very similar, and this is prophase 1. Sorry, I did forget to put that. We're going to call them 1 and 2 from here on out because there are 2 types. Prophase 1 is the first prophase that happens. So prophase 1 is going to look very similar. Now metaphase 1. So in metaphase, we are lining our chromosomes up in the cell. Okay. Whoa. So we're going to have, each of these are called a homologous pair, which is a big word, but it just means that we have, this is dad's replicated copy, this is mom's replicated copy. They're going to line up in the middle of the cell, just like in meiosis or mitosis when we had metaphase. So these guys are going to do it too. They're going to line up. As you can see, it doesn't matter which side red's on, which side blue is on. In fact, it's going to be like shuffling the cards, where you have, it's going to be different in each type of cell. Each meiosis, you'll have metaphase, the blue one's on one side, red one's on another. It's going to be a mix of that, like shuffling the cards. Okay, so metaphase 1, they're lining up, and there's something special that happens in this, in meiosis compared to mitosis. And in metaphase 1 is when it happens. In metaphase 1, these two, so focus on these guys. These are going to do what's called crossing over. The red and blue ones that are touching each other in the middle are going to swap ends of the chromosomes. So we're going to get a little bit of the red on the blue chromosome and vice versa. So now I'm going to have this one that's morphed red and blue, and then this one's got a little bit of red but mostly blue. And we're going to do the same for these guys, just the ends. Okay, so now what you see is every of these homologous pairs, every single one of these is different now. You have this one is entirely blue. This one's mostly blue but has a little bit of red. This one's mostly red but has a little blue. And this one is all red. And same for these guys. So now each of these is slightly different. We call that, what just happened, we call crossing over or recombination, okay? And that's metaphase 1. So this is what you should draw for metaphase 1. Draw after crossing over occurs. And you may want to use colored pencils. I meant to mention this at the very beginning. You may want to use colored pencils because it's hard to represent crossing over without these different colors. So you may want to use the same colors that I am using. Anaphase 1, all we're doing is, they start out like this. We are in at the end of metaphase, but these guys are going to be pulled to this side, and these guys are going to be pulled to this side, just like in our mitosis anaphase. So we have both these homologous pairs are each separated, okay? So whichever's on this side is going to that side, whatever's on that side is going to that side. So that's what you should draw for anaphase 1. Make sure you get the colors right on there. It's important that you have the colors correct. And don't forget to put the recombined chromosome, the ones with the blue and red. You need to show that each one is different when you're modeling this. All right, telophase 1 and cytokinesis. So remember, telophase is when we have the chromosomes are completely pulled up to the opposite side. And then cytokinesis is when we have the cytoplasm and all the organelles split, and then you have a membrane formed between them. And guys, I tried. There is no way to draw this without it looking like a butt. So it just looks like a butt, but that's okay. So we have two different cells now, and this is what we have in each cell. We have one of each of these pairs in each cell. So at the end of telophase 1, we have, instead of 2 pairs, we have only one of the homologous pairs of each type. Okay, so this of chromosome 1, we have only one pair. Chromosome 2, we only have one pair. Okay, so now we're going to begin the next segment of meiosis. We're going to begin meiosis 2. All right, here is prophase 2. This is the beginning of meiosis 2. And it's going to look very similar to the end of telekinesis, or telophase 1 and cytokinesis. So that's what you would draw, is you would just draw that, because all we've done is condense into chromosomes, okay? That's all you need to draw for prophase 2. In metaphase 2, they're going to line up in the middle of the cell, just like they did before with their partner. Okay, so we're lining up in the middle. We're not going to do crossing over again. That only happens in meiosis 1. So crossing over is not going to happen like it did in meiosis 1. So this is all you need to draw for metaphase 2. For anaphase 2, we're going to be splitting these pairs apart and pulling them to the opposite sides of the cell. Okay, that would be anaphase 2. So draw that for anaphase 2. Note that after anaphase 2, they've been pulled into opposite sides of the cell. So this is what's going to be in one cell. This is going to be pulled to another cell. This is going to be pulled to another cell. This is going to be pulled to another cell. Okay, so you can start to see that happening. I'm going to redraw it real quick for you. Okay, now we have it redrawn. This is telophase 2 and cytokinesis. Okay, so now we have 4 cells at the end. And I'm going to bring this up for you without trying not to move the beads. Oops. But you can see for each of these, okay, each of these is slightly different. All right, each of the cells you can see genetically is a little bit different. Each of their colors are slightly different. And that's what happens in meiosis. You get 4 different cells. They are not identical. And it makes sense that they're not identical because this is how you get different sperm and egg cells. So each offspring is slightly different. Why your siblings and you, even though you might have the same parents, are not identical. Okay, so that was walking through modeling meiosis. That's this page here. So you should have this all filled out. Again, if you go back, if you haven't already, do it in colored pencil. Do yourself a favor so you can see the different colors, and each of them are unique at the very end. Then I want you to do this page. So you're going to be assigned for today this page of putting it all together. Okay, so just this page, which is 1 through 8. That's it. That's your assignment today. Tomorrow, we're going to go over these. Big picture from takeaways from these. And then I also want you to watch a video tomorrow because we're going to go over the next one. You notice that you should have this one filled out and do this page. And then you have this other page, which is blank. This one we're going to run through tomorrow. We're going to see what happens when there's a mutation in meiosis. When there's a mutation, what happens? How does that work? And you can see how that mutation can be passed down to offspring. Okay, so we're going to model this just like we modeled this other one. And that will be tomorrow. All right. Let me know if you have any questions. Enjoy the sunshine. It's a beautiful week this week, so try to get outside. Bye.